So I think we can. Okay, so welcome everyone to today's edition of Tissue Talks. Today we are really up to a treat. Our guest and lecturer is Chris Chen. Chris obtained his bachelor's degree in biochemistry from Harvard. He went on to MIT to get master's degree in mechanical engineering and then a PhD in medical engineering and medical physics from Harvard MIT HST department. In parallel, he also earned a medical degree, MD, from the Harvard Medical School. He started his academic career as an assistant professor of biomedical engineering in oncology at Johns Hopkins University, and then was recruited to UPenn as the Shrikanish Professor of Innovation. I think this is very descriptive of what he has been doing throughout his career, the work at the boundaries of disciplines that follows his very inter and multidisciplinary education, and also this first academic title, which is Professor of Innovation. He's now a chair professor, William Fairfield Warren Distinguished Professor at Boston University, where he is directing the Tissue Microfabrication Laboratory. Chris is also the founding director of the Biological Design Center. He's a faculty of the Harvard Wyss Institute for Biologically Inspired Engineering and deputy director of the National Science Foundation Engineering Research Center called CellMet that has played a huge role in our field over the last 6 years. Chris has conducted some of the foundational research in cell and tissue mechanobiology. His doctoral degree was about geometric control of cell life and death. This paper is really a classic. It was published in Science. It amassed over 6,000 citations. And it just opened like a series of papers that followed about cell shape, cytoskeleton, about cell communication through mechanical signals with their environment and control of the cell's fate and function by physical integrations between the cell, among the cells and extracellular matrix. So he's really, he was really one of the people who opened this field that took into account also the physical factor on top of all molecular signals that are determining a cell function. Chris obtained very many awards, and I will just mention the two. He was elected Fellow of the American Institute for Medical and Biological Engineering, and also as a Fellow of the Biomedical Engineering Society. And after that, he was also elected to the National Academy of Inventors. He's also an entrepreneur, and he co-founded and he's co-running a very successful company in tissue engineering space. So it's a real pleasure to welcome him here. And Chris, thank you so much for accepting the invitation. And you can now start sharing your screen.
Okay, great. First, I'm just going to say, as I'm trying to find the sharing, that I'm thrilled to be here. I was just talking to Gordana that I can't imagine when we started this whole, that you guys started this whole Tissue Talk series almost 5 years ago exactly, as everybody was shutting down. And I'm so thrilled that this seminar series has continued to thrive during this time. So I'm really honored to be part of this series. What I'm going to do today is talk to you about some of our latest work in the area. And let me just move this out of the way. What I want to do is just sort of give you an overview of some of the work that we do and some specific projects that I'm excited about. The way I want to start is just asking this very simple question, which is, how do we really make sense of human disease? Like, how do we as a field understand it? And for the most part, almost all diseases that we study, we first study in animals. And I don't mean me, but I mean just the field. And animal models have been extremely helpful in trying to make sense of various types of diseases. But the issue is that there are many species differences between humans and other animal models. And as a result, there are many different types of diseases where we can make some sense of the basic mechanisms, but the actual therapeutic interventions are often not quite right. And that's why so many drugs fail when they go into human trials. Now, as an alternative, we can get rid of the species differences if we simply take human cells and try to study those. And you may know that for the last 100 or so years, there's been this technology to basically chop up tissues and take those tissues and put them on dishes and allow the cells to crawl out. And we can grow and expand many different cell types that we can study that are from people. And that's very exciting. And most recently, of course, there have been these iPS-derived stem cells that can be used to differentiate into many cell types that we couldn't necessarily culture directly from adult tissues. However, despite the fact that those cells are human, when we take them and we put them into dishes, again, they often lose their phenotypic characteristics that they had while they were in your body. They change their gene expression patterns, they change their signaling control systems, and as a result, once again, we're left with a model that doesn't quite represent what we're trying to study. The reason why we think that the cells lose that ability is that they lose the environmental cues that actually tell them how to behave. And so my interest has been in asking whether we can engineer tissue structures in culture using cells. And with that structure, can we sort of fool cells or program them back to behaving like they would while they're in your body? And along those lines, there's sort of two thrusts that I think that the field is very interested in. One is, can we engineer tissues, human-like tissues, for as basically experimental models, organ-on-chip models, things like this, where you could then study, say, a particular disease and gain insights into that disease and develop therapies and test them within these human phantoms, as it were. And the other, of course, is the tissue engineering goal, which is, can you make these tissues in a way that actually makes them therapeutic? You can reimplant them for various regenerative medicine applications. So the question is, if we want to build that kind of environment around cells back to the way that they were when they were in your bodies, one of the things that we always ask is, how do cells normally build tissues? And one of the things that we think about is that when cells are put together and they start to organize into a tissue-like structure, they do so through a constant feedback between their surroundings, the microenvironment, and various cellular programs that are driving, say, proliferation or migration, et cetera. And you can think of it as a slow process where a mix of cells are sort of starting to interact and they generate certain cues that affect the environment, and the environment then feeds back to affect cells. And this eventually evolves into an elaborate tissue. Now, some of those cues are adhesive between cells and their surrounding extracellular matrix. Some are soluble cues. So when cells are producing factors, those factors end up diffusing around and creating sort of paracrine signals between neighboring cells. And of course, depending on where you are within the structure, you maybe can smell certain cell types that are nearby and not others. So again, you can sort of see how the organization of the tissue itself feeds back to affecting how individual cells are signaling within that tissue. And then near and dear to my heart, we're very interested in mechanical forces. And again, the organization of the tissue, whether the cells are embedded in 3 dimensions within that matrix or sitting on a surface, let's say within the bloodstream, they will experience different forces. And one of the things that we're interested in is, again, how do those forces then affect how cells function? Now, you might ask yourself, if this is how the system of cells and tissues actually operates, how do we then study it, perturb it, control it, et cetera? And the thing that my lab has been most interested in doing is developing different types of engineering approaches to basically control the environment around cells, whether that's forces or extracellular matrix or the actual organization of cells using, say, 3D printing. And more recently, we've gotten really interested in using synthetic biology to alter some of the cellular programs that are responding to those environmental cues to drive different behaviors. And so the lab is really interested in developing various types of tools, whether it's microfluidics or various types of matrices, et cetera, to study this process of how cells organize into tissues. And we've mostly focused on vasculature, more recently cardiac, engineering cardiac tissue, and a little bit of work in liver. And I'll show you a little bit of examples from each of those. Now, I'm going to start with talking a little bit about cardiac models. So we have been really interested in taking iPS-derived cardiomyocytes and engineering cardiac tissues with them, again, for the purpose of studying them. There are a number of different types of formats that people have used to study cardiac cells. For example, you can place them as monolayers onto dishes. We feel like that when you do that, you sort of polarize cells in a certain way where there's a sort of a mechanical cues that are coming from one side of the cells. And then there's other things like organoids, and in those settings, the cells are now not on a monolayer, but are in 3 dimensions. But the issue there is that often these cells are sort of unloaded, so mechanical contraction of the cardiomyocytes doesn't feel any resistance to that contraction. And we believe that that affects how the cells behave. And the third sort of type of engineered cardiac tissues are ones that I think of as being sort of loaded. They sort of make a 3-dimensional tissue, but are somehow pulling against some type of resistance. And we've focused mostly on that third type of tissue. Here's an example of one of these tissues that we've made. So it's you can sort of imagine an array of micro wells. They're small wells, maybe a millimeter across. And then inside those wells, we put these 2 pillars made out of PDMS into them. And what we can do then is we can spin down cells with matrix, and when the matrix polymerizes, cells will naturally sort of grab onto the matrix, spread into it, and start to compact it. And when they compact it, the matrix sort of shrinks. And normally they would shrink into a small sphere, but because we have these pillars, the matrix gets caught and sort of bridges these 2 pillars. You notice we put in these little knobs there so that the tissue gets stuck there. And when that happens, then you form this nice aligned structure. Now, one of the things that we've done is we've been collaborating with Gordana, as well as Cricket and John Seidman. Cricket and John are physicians that are interested in studying how different types of mutations in kids leads to the development of different types of cardiomyopathies. So there are cardiomyopathies called dilated cardiomyopathies, where the heart muscle is a little bit weak and the heart sort of balloons over time. There are others called hypertrophic, where the muscle gets thick and the heart almost, the chamber inside sort of shrinks because of that thickening of the muscle. And then there's others that cause arrhythmias. And what they've identified is a number of mutations that are involved in generating these different types of phenomena in people. And what we've been interested in doing with them is seeing if we can model some of those behaviors in our tissues. So shown here is the first study that we did with them, where we looked at kids that had mutations in a gene called titin, which is very important in sarcomeres. And these kids develop dilated cardiomyopathy, where they don't generate the kinds of forces that are needed for heart function. And here we took iPS cells from those kids and from normal kids. And you can see here that when we put them in our system, we can see that the titin mutant cardiomyocytes are unable to contract with the same amount of force that the wild type kids are. And you can see here are 3 different patients with 3 different patients with 3 different titin mutations. They all led to sort of decreased contraction. And then here, what we did was we took a wild type cell line, and using CRISPR generated 5 different mutations, all in titin. And you can see every single one led to, again, contractile defects. So this has gotten us interested in looking at a number of different mutations. Most of the dilated myopathy mutations lead to decreased contractility within the system, and many of the hypertrophic mutations seem to lead to increased contraction. And so we're starting to study what are the commonalities across those. Now, in these cardiomyocytes, one of the really interesting things is that you can study other processes that are going on. One of my postdocs at the time, Anand Chopra, was really interested in looking at the formation of these Z discs. You can see here, these are sarcomeres that are sort of forming across this cell here. And what he noticed is that when he looks at these cells and he trypsinizes them, takes them off of the dish, they lose their sarcomeres. And that's not something that had been really studied before. And so what we wanted to do is ask, why does that happen? And he took a movie of what happens in reverse when you take those cells that are in suspension and you put them back on a dish. And what you can see here is one of the first recordings that he took, where as soon as the cell attaches on the surface, it's starting to make contact, make adhesion points, and you can see that the sarcomeres sort of spontaneously form as a result of that. And you can see here, this is you can almost see the actin treadmilling that's going on inside that recording. We thought that this looked a little bit like the areas where cells might make adhesion with the matrix, so that typically happens in spots. We call those focal adhesions. And there are particular proteins that concentrate in those focal adhesions. One of them is paxillin. So here we labeled paxillin as well as actinin, the marker that labels the sarcomeres. And you can see here in this movie that the sarcomeres indeed appear to be anchored or form at these sites of cell matrix adhesion. As we studied this more, we learned that this ability to generate these sarcomeres requires stress. And that's part of the reason why they need adhesion in order to provide some resistance to the contraction of the sarcomeres. And here I just want to illustrate how important that stress is. Here there are cells that already have sarcomeres formed, and then we added a poison to myosin called blebistatin. And what happens is when you add that in, then myosin loses its activity, and you can sort of see almost immediately that the sarcomeres start to disassemble. So this really led us to understand that there's this important process of sarcomere assembly that requires some level of mechanical stress between the cell's extracellular matrix adhesion points and the sarcomeres that are forming between them. We're also interested in whether adherence junctions between cells, the adhesions between cells, are also important in sort of stabilizing those sarcomeres. And so in general, we think that there's this interesting positive feedback loop between these adhesions, sarcomeres, and forces. Now, one of the things that we've noticed is that in addition to looking at the cardiomyocytes themselves within these tissues, we've gotten interested in the fact that these muscles are multi-cell type. There are other cell types in the tissue other than the cardiomyocytes, and that may end up being important in certain types of disease processes. So just to illustrate, one of those is in hypertrophic cardiomyopathy. You can see here, I told you that the muscle gets thicker in hypertrophic cardiomyopathy, but what you notice is that what's thickening is not more muscle. So even though these HCM mutations often are sarcomeric mutations that cause the cardiomyocytes to contract more strongly, and you might think to yourself, oh, that means the muscle is just getting stronger, like you're Arnold Schwarzenegger as opposed to me in terms of muscle size. But if you actually look at the histological sections, what you see is that in the hypertrophic tissue, what you see is not necessarily more myocytes, more cardiomyocytes. What you see is more collagen and deposition of sort of a fibrotic tissue in between the cardiomyocytes. And so that suggests that there's other cells that are in here that maybe are also being activated in the process of cardiomyopathy. So what we started doing is studying this process in our microtissues, where we have ventricular cardiac fibroblasts as well as our iPS-derived cardiomyocytes mixed together inside our tissues. And what we notice is that in mutations that lead to hypertrophic cardiomyopathy, one of them is this R403Q mutation in myosin, in cardiac myosin, what it does is it seems to cause cells to contract more strongly, but also, if you notice, the tissues become stiffer over time. So if we look at this, here's just sort of a nano indentation of tissues that are made with these R403Q mutants versus wild types. And you notice that these are stiffer. Here you can sort of look at the Young's modulus from this primary data. The other thing that you can notice is that if we stain for collagen, that there's more deposition of collagen in the HCM mutant tissues. And again, you can sort of see that quantified here. We also see that increase not quite as dramatically, but it's still statistically significant for another mutation that causes hypertrophic cardiomyopathy called MYBPC3. Now, these HCM cardiac microtissues are less contractile. So if we look at the sort of beating of the tissue over time, what you notice is that the ones that have these hypertrophic mutations are actually less contractile. And that's a little bit odd because remember I told you that what these mutations do is they actually make myosin work harder, so they should be contracting harder. But our tissues seem to be contracting less. And we believe the reason why is because the tissues are stiffer. Remember, they're more fibrotic, they have more collagen deposition. So as the tissue becomes stiffer, then even though the cells are contracting harder, they're not able to basically deform the tissue itself. We can confirm this where we take those same iPS cardiomyocytes, but instead of having them in a three-dimensional tissue, we just simply put them onto a gel and directly measure how much force they can generate. What you can see is that the 403Q mutant is able to generate substantially more contractile force against its surroundings. So we do know that these cells are inherently more contractile, and even so, the tissues are contracting less. So what's causing this? We took wild type, normal, healthy ventricular cardiac fibroblasts and put them into a tissue with our mutant cardiomyocytes. And what we see is that, in fact, those ventricular cardiac fibroblasts are getting activated by our mutant cells. One of the things that we found is that if we simply cultured them in separate dishes and then took media out from the cardiomyocytes that were hypertrophic and added them into the fibroblast cultures, we saw that it activated the fibroblasts. They started to proliferate more, they produced more collagen, they produced more LOXP, they produced more smooth muscle actin. So they would seem to be getting activated by those cells. And the other thing that's interesting is that if we block stromal cell proliferation in our microtissues, we block that increase in collagen intensity and we block that loss of contractile stress, meaning that the tissues remain soft. They don't get stiffer, they don't get more fibrotic if we prevent the fibroblasts from activating within these tissues. So that tells us the fibroblasts are actually part of the effector that's causing the stiffening of the tissue. And then the other thing that we did was we identified what was inside this media that was causing this. And it turned out there are a lot of factors that are upregulated within these iPS cardiomyocytes that were hypertrophic. And one in particular, EGF, seems to cause the fibroblast to get activated. And if we use an EGFR inhibitor, we can sort of prevent that activation. Collagen production is reduced, and the tissues no longer stiffen in response to that tissue. So this story was just to tell you that in addition to looking at the cardiomyocytes themselves, we're able to sort of model multi-cell type interactions within these tissues. One of the things that we're now starting to think about is how do we organize these cells into larger tissue structures? What I showed you was that we can make these small tissues where multiple cell types are organizing together into an aligned structure. I told you that even at the single cell level within the sarcomeres themselves, they're getting organized as a result of stresses based on where adhesion points are. And so one of the questions that we have is force really an important organizing link across all length scales in organizing the heart? And so we're starting to think about how to study that. along those lines, one of the things that I got really excited about in organizing was this engineering research center that was funded by NSF. We're nearing completion of this project, um, where what we're trying to do is really study this process of, can we take various engineering tools from across all of our colleagues and use it to better engineer heart tissue for a variety of applications. So this involves BU, University of Michigan, Florida International University, Harvard, Columbia, and many other folks, trying to think about how do we take engineering approaches and apply them to engineering heart tissue. What I want to share with you is just one example of something that we've done in this space with collaborators. There's one thing that I've been sort of frustrated by with microtissue models, and that is that the mechanics of contraction in these microtissues is quite different than the mechanics of real heart. And just to kind of illustrate that, you know that these kind of act like cantilevers, so they sort of act like a spring, and you can sort of think of the spring deflection sort of is linear with force. That's just like if you were to sort of exercise by pulling against a spring. But the heart doesn't generate those kinds of forces or experience those types of forces. What happens is something more like this. If you think of sort of, again, pressure as being like a measure of stress and volume a measure of strain, what happens is that the tissue is relaxed, the heart is relaxed when it fills, right? So it gets stretched in this relaxed phase. Then you activate contraction and it starts to increase its contraction up to a point where you reach a pressure that exceeds the pressure of blood in the aorta. The aortic valve opens, and then the heart contracts in this sort of high stress state until it reaches a point where it's finished its contraction cycle and relaxes. So what you can see is that the loading and unloading periods experience very different stresses. And the reason why that's important is that if you load the heart at different parts of the cycle, you get different types of remodeling that occur. And so we'd like to be able to study those two processes separately. And in this kind of model system, we can't do that. So one of my students connected with two other professors, Alice White, who's an expert in 3D printing, and Arvind Agarwal, who's a mechanics engineer expert. And so what they did was they asked, well, could we make a chamber that can deform and make it soft enough that it can deform in response to cardiac contractions? And so what they did was they built this system where they made very tiny struts using a 2-photon laser direct writer. And then you could sort of adjust the design of the cage so that you can adjust the mechanics of it. And so using that kind of approach, then we could sort of tune the mechanics of the contraction of the cardiac chamber. What we did was we basically put that into a well, and just like earlier, we can seed cells with matrix around it. The cells will then compact into a tissue around these chambers, and then they will spontaneously contract, just like I showed you earlier, except now they're contracting to sort of expel fluid out of this chamber. And then you can sort of see this is a recording of fluid with beads outside of the chamber. Now, one thing you'll notice is that, of course, there's a single exit, right? And so it's still symmetric, meaning it contracts and sucks in fluid with the same mechanics. There's no asymmetry here. To develop that asymmetry, what you need are valves. And so using that same laser writer, what Christos was able to do is within the microfluidic device that he set up, he's actually able to write these valves inside the microfluidic chamber as he's built it. And so you can sort of see here, this is him sort of applying pressure on either side of the valve to either open or close it. And then over here, you can sort of see what happens when you put that valve inside a system that has, again, fluid moving back and forth. But with the valve, you can sort of rectify that flow. So what he's done here is now taken this cardiac chamber, it's generating pumping motion, and he put in this microfluidic circuit where there's two wells, and in one well he's put in a one-way valve so that fluid can go from the well through that valve and then down in to fill the cardiac chamber. But when the cardiac chamber contracts, this valve closes and fluid instead goes out through this one-way valve into this well. Now, by doing that, what it allows him to do now is pressurize the intake pressure versus the outflow pressure to be different levels by simply putting in different amounts of fluid into these two different wells, so you have a gravitational difference. So using this method, then you can actually see at different points within the channels that you're able to sort of move fluid and rectify basically this process. So using that now by putting a higher fluid load over here on the exit side versus the inside, then you get a filling at a low pressure, then it contracts and it has to exit at a high pressure. So this to us feels much more like the sort of normal cardiac cycle. And we're really excited about this because now we sort of study the mechanics of what the stresses are across these two different settings and their impacts on cardiac function. Now, what I want to do is switch gears and talk to you about sort of a second arm of my lab. We're really interested in engineering vasculature in addition to engineering cardiac tissue. And I wanted to share with you some of the work that we're doing in that space. So we have lots of different model systems that we engineer vasculature. We have ones that are sort of pre-designed, sort of specified vascular structures, very simple channels, et cetera. We have ones where we're studying angiogenesis or vasculogenesis, where cells are actually remodeling to form new vascular networks. And we do some work in trying to think about how to build vasculature to enhance vascularization in vivo. And so I wanted to just give you some illustrations of work in these spaces. So engineering functional vasculature, one of the things that we're interested in doing is studying basically the mechanics of the vasculature. You all know that shear stress and hoop stresses from pressure have important effects on the endothelium that lines those vessels. And so one of the things that we're doing is just studying those forces and how they affect the endothelium within them. So here's one example of that, where what we've done is we've made a channel. Just think of this as this red area, sort of like a collagen gel. We can have a channel that goes through that gel, cylindrical channel. We can seed it with endothelium. This is what the endothelium looks like. And then we can apply a known fluid flow through that channel to study the effects of shear stress. One of the things that we observed, which is known, is that shear stress is quite important to cause endothelium to form a good tight barrier between the blood side and the tissue side. And you can see that illustrated here. We flow in a dye, fluorescent dye, and if we have no flow in the system, then the dye leaks out very quickly. If we apply a certain level of shear stress within the system after a couple of hours, the endothelium becomes very tight to that leakage. And you can see here images of what the cells look like when they're in this sort of static culture versus an underflow. And you can see how this flow seems to drive really nice cell-cell junction formation within the endothelium. We got really interested in trying to understand what is causing cells to sense that shear stress and how do they turn that sensing into a change in cell-cell junctions. One of the things that we discovered was that shear stress activates a particular receptor called Notch. And Notch is really interesting because what Notch does is it gets activated by cleavage. So what happens is it binds to a ligand called Notch ligand, like in this case DLL4. And when the two are bound, then what happens is that there's a protease that will cleave this extracellular domain of Notch away. That causes a conformational change in the rest of the receptor that causes another protease to cleave the intracellular domain away. Now, what's interesting is that what's known about Notch is that intracellular domain is a transcription factor that turns out to be really important for arterial fate of endothelium. So the ICD, this intracellular domain, helps keep arterial fate within the endothelium. What we discovered is that there's this extra little piece that's left inside the transmembrane called the transmembrane domain. And that transmembrane domain turns out to be the signal that drives cell-cell junction assembly. What it does is it activates RAC. RAC causes actin polymerization and locks V-cadherin, the adhesion junction receptor, to basically assemble at the cell-cell junctions. So that work was published many years ago. And what we've been doing recently is studying how that's related to another set of factors that are really important in controlling the barrier function and health of endothelium. And that is lipids. There's a number of lipids that cause your endothelium to become more inflamed. And then there's other lipids that are good lipids, quote unquote, that prevent your endothelium from becoming inflamed. So one of those good lipids is called sphingosine 1-phosphate. Sphingosine 1-phosphate is the major bioactive lipid that is in HDL. And if you remember, HDL is good, LDL is bad, cholesterol can be bad. So it turns out that this S1P, what it does is it causes barrier function by causing cell-cell junction assembly, just like the way shear stress does. And what we've been studying is there's an interaction between S1P receptor, which is a GPCR, and shear stress. And so that is work that is not yet published, but we're working on it right now. One of the things that we've been really interested in doing is taking that model system that it sort of talked about earlier, shear stress and its effect on endothelium, and thinking about it in the context of patterning, like how do you form complex vasculature? So what I showed you was a really simple single channel. What we'd like to do, for example, is make bifurcations. It's well known that in bifurcations that there's an increased predilection for atherosclerotic plaques to form, and that's because there are recirculating flows at these bifurcations that don't occur in sort of a smooth channel. The same is true in aneurysms, and the same is true in malformations. Now, the thing is that I would love to be able to make these things, but I couldn't make them. Like we could make our single channels by just taking a single needle and sort of sliding it through a gel, polymerizing the gel and pulling the needle out, right? So you can make a nice smooth cylinder. But we don't know how to make these types of structures in a highly specified way. I was complaining about it, and one of my very talented postdocs came up with a solution that I'm really excited about. So there have been some work from a number of folks, including Jennifer Lewis's group and Jordan Miller, who was a former postdoc of mine, his group, looking at different ways of basically using 3D printing to be able to build complex vascular structures. And they work really quite well, but they're sort of restricted to sort of a certain size scale, mostly in the sort of 100 micron to 1 millimeter scale. And it's very hard to build multi-scalar structures, meaning structures that have both big vessels and small vessels all together. And part of the reason why is because it's hard to fabricate at different length scales altogether. And what my postdoc, Subra, had thought is that, well, we have many ways of making very complex structures with many length scales in them, like your iPhone. It's just that we can't do it with biological materials. And so what he asked is, can we just take advantage of the existing fabrication processes and then use like a molding technique to sort of switch from a non-biological material to a biological material as a way to solve this problem? And the key challenge is how do you transfer that pattern? And so what he did was he came up with this idea where you take some pattern that you've already fabricated in some way, and then you can make a mold of that we know how to do. You can use PDMS or other things like that. But the next piece is how do you make a mold of that mold? And what he did was he used a metal called gallium, which molds very nicely into the structure. And metals can mold at very high resolutions. That's why metals are the preferred molding method. But what's special about gallium is one, it's non-toxic, and two, is it melts basically at room temperature, 28 Celsius. And so you can transition between a solid and a liquid, which means that you can make the structure, you can pour a nice hydrogel around it that might be quite soft, and then instead of trying to rip that mold out, you can actually melt it so it sort of extrudes itself out. And so this is an example of something where he used the Stanford bunny, made a structure, molded it through this process, and at the end seeded cells into the basically gaps inside the structure. So here, this is one of the examples of how this works. So here you have a single channel that's made out of this gallium metal. And what he does is he simply transitions it from solid to liquid And one of the things that you might imagine happens when it transitions to liquid is this gallium has high surface tension. It's like mercury. It might just ball up right in the middle of the gel and sort of rip the gel. But it doesn't do that. And the reason why it doesn't do that is that he's created an asymmetry in the system. You notice here it's a little thicker than over here. What that does is it causes a difference in basically the surface tension that's created here versus here. And what that does is it creates a pressure difference. There's a higher pressure here than here because of that radius of curvature is smaller here. So that causes the gallium to then push itself out in this direction. And you can see that in this still, and then in the same movie here, you can sort of see what happens is it just sort of draws itself out without causing any damage to the gel. So that's interesting. That's a sort of single channel. Of course, we can do that with a needle, but there are many things that we can't do by pulling something out of a gel. And here's an example where he's now made sort of a branching structure. You can imagine that if you had this structure and you molded a material around it, like collagen, there's no way to get this tree out, right? You can't just sort of pull it out. It would rip the gel. And so what he does is then liquefies it. And again, you can sort of see here the fact that this side has sort of a larger branch means that a higher curvature here will create high pressure and push the gallium out of the tree. And you can sort of see that in this movie here. And you can see that once it pulls itself out, there's no damage to the surrounding hydrogel that was formed. And then we can backseed endothelial cells into that, and it sort of seeds down into these structures. So we think that this technique could be quite useful for making fairly complex structures. I had asked him in the first place to do this so that we could study aneurysms. And so now we're starting to make these sort of different types of sizes of aneurysms with different sort of extents to which that aneurysm is sitting out or embedded into the channel. And you can see that you can mold them quite nicely. Again, if you had a needle like this, if you pulled it, you would rip them. So here, when it melts, it sort of these things just sort of fall away into the main channel. So we're really excited about using this technique now to study how the endothelium now reacts to this geometry, both in a static setting, but also under flow. And it's something that I'm really interested in studying. He did some other crazy things. So like here, he made a grooved, sort of like a screw. And again, that's something that you can now melt away, but what's left behind are grooves in the collagen gel or fibrin or whatever you want to mold. And when he seeds endothelium, you can see that it aligns within that structure. So we can sort of now study, for example, how alignment of endothelium affects its sensitivity to shear stress. And of course, you can make non-endothelial structures. So here he molded sort of a structure that was based on alveoli. So we think that this kind of technique could be used to make other types of complex 3D structures. The last thing I want to talk to you a little bit about is some of the work that we're doing, not so much in molding and making sort of specified material vasculature, but how do we get cells to form vasculature that is potentially useful? And what we've been doing over the years is using biomaterials and architecture and growth factors to drive angiogenesis and to ask what is the interaction between these different cues to get endothelial cells to form the vasculature that we want to. And there's some progress, but there are also some limitations. So for example, we started in this area by using 3D printing to make sort of different vascular channels within a construct and then trying to implant it. And one of the things we observed is that by doing so, we're able to actually pattern or guide how vasculature forms. So for example, here, we made these sort of parallel channels. We filled them with endothelial cells. The endothelial cells help to guide new endothelial cells from the host to sort of anastomose and connect with those. You can see here, if we take a gel that's just seeded with endothelium that forms a vascular network and then implant it afterwards, they connect with a host vasculature. We can then inject a fluorescent dye into the blood, and you can sort of see the perfused vasculature that's formed, and you can see it's sort of tortuous and not very well organized. So there's flow in here, but it's not very good flow. In contrast, if we make these sort of channels, then what we see is that we get a capillary bed that's formed that's a lot more organized, and we get better flow through these structures. Here you can see my grad student got a little cute and sort of made a channel, implanted it, and was able to find sort of that same channel inside the construct later. And you can see here, this is the host vasculature. These are our engineered vasculature, and they do somehow connect and are able to get good perfusion this way. So this is quite exciting. This is something that we're using to try to improve vascularization of certain types of implants. One of my postdocs got really interested in a question that was sort of inspired by this, which is, can we control the extent of vascularization of something? Can we make something that vascularizes more or vascularizes less? And that seems like something I think is quite hard to do. And we think that that's quite interesting because different tissues have different amounts of vasculature. So shouldn't we be able to tune how much vasculature is generated in a particular setting? And we certainly know that, for example, as you age, you have different extents of vascularization within those tissues. And so we'd like to be able to tune that specifically for different situations. So the idea that my postdoc Mai came up with, she's now at University of Wisconsin, and Mo Khalil, one of my colleagues who's in the Biological Design Center and a synthetic biologist, was could we actually put VEGF or other types of growth factors that we know control vascularization, can we put them in a cassette that we can put into cells where we can tune the extent of vascularization using an external small molecule? So in this case, what Mo did was he had engineered a synthetic transcription factor that gets activated with different concentrations of this FDA-approved drug, ruzasvir. And what he shows is that you can get more gene expression with more of this small molecule. And what Mai did was she took that promoter and put VEGF onto the cassette so that we could produce more VEGF with it, and then asked, could we use this in different settings? So the first thing that she did was she studied this in the context of an in vitro model that we have, sort of an organ-on-chip model, to sort of see whether we can control the extent of vascularization. And here, what you see that she did was that by increasing the amount of ruzasvir, she can basically tune the amount of VEGF that's being produced by fibroblasts around endothelial cells, and then the endothelial cells get activated and start to form a more dense vasculature. We can also show that in our system that we can actually apply flow through these vascular networks and get perfusion. You can see that in this blue dye here. And what's interesting is that depending on the amount of ruzasvir that you put in, you can sort of tune the density of vasculature that you form. And so this is a project that's still ongoing. It's not yet published, but it's something that we're excited about because you can imagine that there's certain settings where you need more or you need less, depending on the situation, and you'd like to be able to sort of tune that on demand. Shown here, just our first work where we've implanted some of these cells into mice, and you can see again that we can sort of induce vascularization, and we're now doing some work to sort of characterize that process. And then the last thing I want to show you is that we've been collaborating with Sangeeta Bhatia for many years. She's a classmate of mine from grad school, and she works in liver tissue engineering. So we've been taking these same vascular structures and implanting them now with liver structures and showing we can get good vascularization of these constructs. But what I wanted to share with you is that another grad student of mine got interested in this problem that although we can implant these tiny little constructs, there's really no way to think about how we do that in the context of a human, because we would have to generate very large structures. And if we generate those very large structures and we try to implant them, they would inevitably become hypoxic in the center of those structures. And so what she thought was, well, what if we could implant small structures, but then trigger them to grow after you've implanted them? And so she asked, this is Amy, one of my grad students, can we sort of hijack the normal regenerative programs that cause liver to grow, but we turn them on when we want to after we implant the hepatocyte structures. So what she's done is basically found different factors that are important for causing hepatocytes to grow from liver regeneration literature, and then asked if we install them into fibroblasts or endothelial cells in our implant so that they'll produce these factors after they've been implanted. Can we do the same thing I showed you earlier with the vascularization, but now trigger hepatocyte growth? And so she's now just getting this wrapped up. And what she's shown is that there are certain combinations of growth factors that upon implantation and then triggering the expression of those factors, we can cause the hepatocytes within the tissue now to start to expand locally. And with that increased expansion, we also see increased vascularization, et cetera. So we're quite excited about this strategy as sort of another approach to sort of engineering tissues that's not just focusing on the microenvironment around cells, but also thinking about how to control the cells themselves. So with that, I just want to say that I hope that I've given you some thoughts about how we think about how to engineer functional tissues. And just wanted to say that I appreciate your attention. One of the things that we're really interested in is this whole idea that in order to really understand these different kinds of model systems, I feel like we really need better and more sort of interaction between biology and engineering, both from a people standpoint and department standpoint, and field standpoint, training standpoint. And only then are we going to be able to really figure out how to make more sense of these complex biological systems. And then the second message I wanted to sort of convey is that we really feel that in vivo studies will always be there, and traditional sort of reductionist mechanistic studies will always be there. But we really feel like these, I'll call them organotypic models of trying to build more complex tissue functions within cultures, is really an important platform that I think is going to change the way that we do science over the next century to better understand these systems. And with that, I just wanted to thank you for your attention. I think I've shown all of the people that were involved in the various projects along the way, but as you know, this is all only possible by a really incredible and innovative team. So I want to thank my postdocs and grad students for their efforts in this work.
Yes, thank you. This was really amazing. And I also like the ending about this big convergence of concepts and disciplines that are likely to continue to drive the progress. So thanks very much. So we do have time for a few questions. Let me just say that our next speaker next week we will be Kerry Athanasiou. We don't have the title yet, but he does work in orthopedic space. So this will be a different story about a completely different organ system. So you got quite a few questions. Maybe we can start from the one asked by Natalie Pachter. It's about the heart tissue, and she is interested in the role that activation of supporting cells of fibroblasts may play, that activation itself may play in the concepts that you are developing, and if you looked into it, and if you expect that the activated versus non-activated fibroblasts may make difference in tissue development and vascularization.
Yeah, that's a really great question. So as Natalie probably knows, culturing fibroblasts in vitro is very challenging, and that's because the presence of serum or other such factors can itself activate fibroblasts, and certainly culture on dishes does that as well. And so we do our best to try to use fibroblasts that are not activated when we start our cultures. And I think that the presence of the hypertrophic cardiomyopathy mutations, we believe, is causing inflammation within the cardiomyocytes themselves, which causes them to release these sort of inflammatory cytokines that then activate the fibroblasts. But it is true that fibroblasts in our hands as well as everybody's are never quite the same in culture as they are in your bodies, never quite as quiescent. So it is something that one has to think about how to deal with.
Okay, so maybe we can take another one or two questions. Question by Stephen Adebayo. If you could make a comparison, oops, sorry. I lost the question for the moment. Okay, so which markers can facilitate mechanotransduction in cardiac tissue development?
Which markers can facilitate mechanotransduction?
I guess which factors probably.
Yeah. So there's certainly different uh, um, conditions that we think are important in the context of getting cardiac cells to be more contractile, for example. So Gordana, for example, has shown that electrical activity and sort of pacing seems to help these cardiomyocytes to mature and generate more force, uh, change their gene expression patterns to be more mature. We think that contractile activity and the sort of sensing of that resistance as they're contracting more is probably part of the autocrine loop that makes the cardiomyocytes become more mature. So if you were to increase pacing, but the cells were suspended and not feeling any forces, they probably wouldn't mature in the same way. So I do think that there's a sort of interesting interaction between cell matrix interactions, the tissue itself, the resistance to forces that are all sort of feeding into what makes a cardiomyocyte more mature. I think you also maybe are asking a little bit about development. The developmental processes in early heart formation is very complex. You know, that the heart starts as a tube and sort of has to fold around in order to form these different chambers. And we, as a field, know very little about what are the sort of mechanics of that process of folding and forming those cardiac chambers. So I think the field is still wide open as to what are the things that matter there.
Okay, and maybe Chris, to close with a like a very, very broad question that Rosemary Hunziker is asking. She's essentially asking if you envision possibility and also benefits of raising collaboration to the level of large, already established entities. So she's mentioning CellMet that you are serving as a scientific lead of as one, and then there is also Tissue Engineering Research Center. So can we sort of converge also in these other levels? And if so, like what may be the benefits that such interaction may result in?
Rosemary always asks
Yeah.
the hardest yet simple, appearingly simple questions. So I would say absolutely 100% that at the meta level, it's really important having centers and sort of groups of scientists working together to sort of chase some of these bigger problems. In my experience, what we were doing as a single lab, you sort of tend to focus on projects and problems that a single graduate student is able to sort of achieve at any one time. But as a center, you're sort of trying to get multiple people to align their work so that you can go after the bigger problems that you wouldn't be able to do as a single lab. You wouldn't want to take the risks to do that. And I really think the centers provide that platform in order to chase the bigger, more meaningful steps forward in the field. And you can see in this field, for example, there are obvious, very big problems that need to be addressed that probably not a single graduate student could address, right? You need sort of these bigger efforts. And I do think that funding agencies need to think about how to enable us to be able to organize in that way so that we're able to collaborate more. And I really enjoy those collaborations because it allows teams that have really different expertise to come together and really have fun trying to chase some of these important problems together.
Yes, thanks so much. We do need to let you go, and I hope that you will agree, like other speakers, to answer the remaining questions offline. We will provide you with the contact information of people because there is a long list that we just couldn't
Yeah.
make it through. Thanks everyone, and see you next week.
Thank you so much, and happy to answer questions by email.
Okay, thanks, Chris.